We're going to talk about RFLPs and VNTRs. Ever heard of those? We're going to find out what they are. But first of all, does anybody know who that is? Let me enlarge that picture a little bit. No? Well, no. Anybody know who this is? Recognize the face? Justin? No? His name is Dr. Flaviano Giorgini. He is a genetics professor. That means PhD researcher. He also does teaching, research and teaching, in England at a university called Leicester University. It looks like it's spelled Leicester, but it's actually Leicester. It's pronounced Leicester. And he does genetics research. And I found him online, and I thought it was really interesting because he's doing some work with neurodegenerative diseases. Diseases that affect the nervous system, that break down the nervous system over time. Remember Huntington's disease, you guys? You remember that one? Terrible genetic disease, autosomal dominant. So we learned about it last semester. That's the one that strikes at onsets in middle age. Well, different people have different opinions on when middle age starts. Okay? Some people with the gene, and it's a dominant, autosomal dominant gene. Some people with the gene experience the onset. In other words, the disease starts around age 35, some 40, 45, 50, 55. It can vary when it hits. But their life is totally normal up until that day. And then all of a sudden, they forget where their keys are. I mean, that happens to us all the time, right? But they slur a few words. All of a sudden, on one day, that happens, and it starts. But then, if that person has the gene, from that point on, it may be 7 years, it might be 10 years, slow downhill slide, ending in death. But during that 7 to 10 year time period, they're going downhill mentally and physically. Well, and emotionally, as you might imagine. Because what's happening is brain cells are dying off, and they go from being normal to starting to kind of shake a little bit and have trouble controlling voluntary muscles. The old disease, or the old name of the disease, was Huntington's chorea. Chorea, C-H-O-R-E-A, as in like choreography, dancing movements, because as the disease progresses and they lose control of their voluntary muscles, they start kind of moving uncontrollably, like, you know, kind of dancing, kind of jerking movements. But then they go from normal to having to use a cane, and eventually a walker, eventually a wheelchair, eventually they're bedridden and paralyzed, and then they die. It might be 7 to 10 years. Now, at the same time that they're losing their physical abilities, they're losing mental abilities as well. Okay, at the same time. So they're going from having normal intelligence to mild mental handicaps, downhill slide into moderate, eventually severe, finally profound, as extremely mentally handicapped as you can be. Now, at the same time, and this to me would be the hardest part, at the same time they know what's happening to them. Okay? I mean, so how would you feel if you knew that within 7 or 10 years you would eventually have the intelligence of a 1-year-old. And towards the end, somebody would have to feed you, change your diaper, bathe you, and all that stuff. So the patients who have Huntington's disease, they suffer severe depression as well. Okay, so that's the nature of the disease. So if you think about it, every gene in their genome is working just fine except one. Autosomal dominant. That's Huntington's disease. Well, this researcher, Dr. Flaviano Giorgini, I was interested in what he's doing because one of the diseases that's being researched in his lab is Huntington's disease. And I thought that's pretty cool because that's kind of what we're talking about today. Now here's the other thing that's interesting about this PhD researcher here. It says, I could not believe this when I saw this. It says he grew up amidst the cornfields of Indiana. And he got his bachelor's degree at Purdue University. That's just across the river. He's a leading research focusing on neurodegenerative genetic diseases, especially Huntington's disease. Got his bachelor's degree in biology at Purdue. Then he got his master's, it says, at Washington University in St. Louis. And then his PhD at the University of Washington in Seattle. So now he's a researcher in England, Leicester University. I thought that's really cool. Oh, there he is up close and personal. Look at that. BS degree, West Lafayette, Purdue. Go Boilers. And here he is with his lab members. Okay, there he is. And these are all, some of those are probably master's degree students doing research in his lab, or students, graduate students working on PhDs doing research in his lab. Those are the members of his lab at Leicester University in England. Now, that's him back in high school, 1988. Jefferson High School, Lafayette, Indiana. He was in my genetics class back in 1988. Right here at Jeff. That's him right there. You recognize him? That's him. That's kind of neat.
Hey, there's you.
Oh, that's me. That's me back in 1980. Oh my gosh, my hair was brown. Now there's some gray in there. See, there's some gray in there. Those are highlights. That's gray hair. Yeah, that's me. I was young.
What did you teach about genetics in 1988?
Great question. What did I teach about genetics in 1988?
Because most everything we learn is like...
Well, almost everything, well, I'll tell you this. Everything we're learning in here this semester was not discovered when I graduated from college. Not discovered yet. So everything in here that we're learning this semester, I've had to learn since I graduated from college. But I graduated from Purdue in 1977. This was 1988. So we knew a little bit more in 1988 than we did in 1977. Okay? But a lot's been learned since 1988. So the course is very different than it was then. But this was, I was coaching our school's Science Olympiad team. That's what this is. And look at the medals that they won. I was so proud of them. I was so proud. Yeah, we had this Science Olympiad team, and I think this was at some school in Indianapolis where there was a big meet on a Saturday where all these schools competed. It's kind of like the Olympics. Science Olympiad is a little bit like the Olympics, and there are all these competitions. And so different students on the team were in different events. Like this girl, for example, right here. She was a foreign exchange student from Germany. And so I put her in an event called metric estimation.
You trick.
No, that's just good coaching. That's not tricky. That's good coaching, right? Yeah, metric estimation. I remember that was an event where, like, you go into this room the students would go into this room and they'd have like maybe a big box sitting here on a table, and that was one of the stations. And you had to look at the box and estimate like how many cubic centimeters does that box hold? Like what's the volume of that box in cubic centimeters? Or here's something that's this tall. Just looking at it, you have to estimate like how many centimeters tall is that? That kind of thing. Metric estimation. So why did I put her in that event?
Because they do metric.
They use metric system in Europe, right? Right. And she got the gold medal, so that's pretty cool. It's not cheating, right? It's good coaching, right?
Have you reached
Good.
out to that guy? Say, I'm
Huh?
a high school genetics teacher. Congrats.
Say what?
Have you reached out to that guy?
Have I reached out to him? Oh yeah, I've had coffee with him at Panera's when he was in the States. I managed to meet up with him. Now what's really interesting is the guy standing next to, oh, look at this. Here's a life lesson. Do you see any medals hanging around his neck? He didn't win anything. I mean, he was a really good student, but he didn't win anything. So I think the moral of the story is, high school is not the end of your career, right? It's only the beginning. I mean, now he's doing some great things. So, but the guy standing next to him, this guy here, Rick Harris, I haven't heard from him in decades. And I got an email from him yesterday. I couldn't believe it. You want to know how I was doing? It was pretty cool. And he sent me a paper about something about the effects of magnetic fields on regeneration of planaria. Those are little flatworms. And some researcher has been doing this research on the effects of magnetic fields on regeneration. You know, like little planarians, a little flatworm, you cut it in half and each half can grow a new half. That's called regeneration. And he was all excited because he said, look what research is being done now in 2019. And that's kind of what he did for his science fair project back in 1988. So it was really cool kind of reconnecting with him. But he's a professor in the medical school at the University of Michigan up in Ann Arbor. So he teaches medical students. So that's kind of cool. So you never know what people are going to do, you know, when they grow up. So, yeah. Gosh, I was so young there, right? Do I look like a kid? Kind of? Sort of? Kind of? Oh, Dr. Flaviano Giorgini. This was in the Journal and Courier back in 2015. Sometimes he'll come back to the United States and visit. He's got a brother here who lives in Lafayette. There's his brother. Those were the Giorgini brothers. Sounds like a flying trapeze act, doesn't it? The Giorgini brothers, Massimiliano, we called him Mass. Flaviano, he went by Flav. And I had them both in genetics class back in 1988. But Mass is quite an accomplished musician. Actually, these guys had a band, a punk rock band, back in high school called Rat Tail Grenadier. That was back in the days when a lot of guys had like what looked like a regular normal short haircut, but when they turned around there'd be this rat tail hanging down from the back. That was in the late 80s. You ever heard of that? Ever seen pictures of that? So their band was called Rat Tail Grenadier. Now I don't know if that's still the name of their band, but they still get together once in a while. They have gigs just for fun. There he is, the PhD geneticist right there. And that was in the Journal and Courier because they're local hometown boys. That's their band. Kind of fun. So I thought that was cool. All right, DNA fingerprinting. Take your study guides and take a look at page number 6. Now, I was proud of you guys the way you worked in that lab last Friday and Monday when you ran the DNA fingerprint gels and you did the electrophoresis. And that's sort of how it's done in the real world. Sort of. There are a few more things we would have to do to that lab if we wanted to do it exactly the way they do it in the real world. But there's a couple different ways of doing DNA fingerprinting that I want to get into, and that's right here on page 6. Now, here's the guy who invented DNA fingerprinting. His name is Alec Jeffries. Back in the early 1980s, 1984, he's at the University of Leicester. Sound familiar? In Leicester, England. And that's where Flav is right now. Flav Georgini. That's where he is. Small world, right? Okay. On top of page 6, the title here, RFLPs, stands for Restriction Fragment Length Polymorphisms. That's a mouthful, isn't it? RFLPs. Now, people who work with this stuff all the time, they don't usually walk around talking about restriction fragment length polymorphisms, because that's a mouthful, right? I mean, it takes a lot of energy to say restriction fragment length polymorphisms. So usually they just say RFLPs. And some people even shorten it and say RFLPs. So if you ever hear about RFLPs, Daquan, that's what we're talking about. RFLPs. Don't you agree that's easier to say than restriction fragment length polymorphisms? Okay, I think we know what that term means. Restriction fragments, what are those? Raise your hand if you know the answer. What are restriction fragments? If nobody raises their hand, the random pointing finger of doom will pick somebody. What are restriction fragments? Yeah.
Small pieces of DNA.
Yeah, those little tiny pieces of DNA that you get after you cut the DNA up with a restriction enzyme, right? Restriction fragments. Okay. Length, we know what that word means. Polymorphisms. Anybody know what that prefix poly means?
Many.
Excellent, means many. Morphisms, shapes or forms. Polymorphisms, you should probably write this down. Polymorphisms. A polymorphism is a trait, or it could be a piece of DNA that varies from person to person in a population. A trait that varies a great deal from person to person across a population. We call those polymorphisms. Can anybody think of a trait? A trait in humans that is really different from one person to another and varies a great deal within a population.
Hair.
Excellent. Hair color. Hair color. Yeah, some people are really light blonde, some are kind of blonde, some are sort of blonde, some are really blonde. Some are light brown, dark brown, medium brown, auburn, red,
White.
white. Thanks a lot.
You have to be inclusive.
Right. I feel so warm that I'm included in that group. Black, really black, sort of black, kind of black, jet black, all kinds of shades, right?
Don't forget about gray.
That's right. So hair color is highly polymorphic, right? Oh, how about fingerprint patterns? No two are the same? That's a trait that's highly polymorphic, okay? Another trait that's highly polymorphic, the restriction fragment patterns on gels of people. When you do DNA fingerprints, if we did everybody's DNA fingerprint in here, we would all be totally unique, right? DNA fingerprint patterns are highly polymorphic. So, restriction, fragment, length polymorphisms. Basically, differences in the DNA fingerprint patterns among different people. No two are alike. Highly polymorphic. Restriction, fragment, length polymorphisms. Make sense? Now, there are two different kinds of RFLPs. The first one, restriction site polymorphisms. Restriction site polymorphisms. Differences in the location of restriction sites. That's what you guys did when you did the lab earlier this week. If somebody ever asks you, oh, you did DNA fingerprinting in genetic class, what kind of DNA fingerprinting did you do? This is what you tell them, Angie. We did DNA fingerprinting based on restriction site polymorphisms, and you'll impress them mightily. Okay? Opportunity to put on that nerd swag.
Oh, down.
Yeah, that's cool. We did DNA fingerprinting using restriction site polymorphisms. Differences in the locations of restriction sites between different people. Wait, time out. What's a restriction site again? What's a
Place
restriction?
where the restriction is not cut.
Excellent. The place where a restriction enzyme cuts. Different people have different restriction sites, remember, at different places within their DNA. And that generates different numbers and lengths of restriction fragments in different people. And so when you run their DNA through a gel, you get different patterns that are unique from one person to another. So just as a little bit of a review, because I know you've slept since then, here's how that works. Let's say, okay, this is person A, and we're looking at this sequence of DNA from here to here. And person A has a restriction site right there, where that arrow is. Let's say that we've used my favorite restriction enzyme, echo R1, that cuts wherever it encounters GAATTC. So there's a GAATTC in the DNA right there. So the enzyme cuts right there. And so when we run person A's DNA through a gel, we get this really long fragment here and this short fragment here. There's the long one, there's the short one. You get two bands in the gel like this, right? That makes sense? That's kind of what we did in the lab. Person B, on the other hand, is a different person. And so she's got a GAATTC here and here. So she's got two cut sites in the same sequence that we looked at in person A. She's got two cut sites, generating 1, 2, 3 fragments. You with me? 3 fragments. So when we run person B's DNA through a gel, we get 1, 2, 3 bands. That's kind of what restriction site polymorphisms looks like. Okay. Make sense? Now you see, in this second cut site right here, maybe person A at the exact same place doesn't have a GAATTC. Maybe he's got a GAATTA. So the enzyme doesn't recognize that, doesn't cut there. Just one little base difference between that person and that person. Okay. All right. That's the kind of DNA fingerprinting that we did in the lab, based on restriction site polymorphisms. Now, take a look at page 7. There's another kind of RFLPs called, there's a mouthful, variable number of tandem repeats. VNTRs. Variable number of tandem repeats. VNTRs. Now, if you wander around the halls of Jeff High School and you hear students talking about VNTRs, that would be really weird, wouldn't it?
A little bit.
If you hear the same students talking about mini satellites, they're talking about the same thing. Mini satellites, that's just another word for variable number of tandem repeats. VNTRs. Variable, what's variable mean? If something is variable, what's that mean? If something's variable,
Not all the same.
yeah,
Random.
all different. If I tell you that I'm looking down the halls during passing periods and I look at people's heights, how tall people are as they're moving through the hallway, and I say, boy, the heights of all those people, that's quite variable. In other words, no two exactly alike. They can vary a great deal. Variable. Number, we know what that means. What's tandem mean? Does anybody know what that word means? Tandem? Have any of you guys ever ridden a tandem bicycle?
2 person bicycles, yeah.
Yeah, a bicycle built for two? Raise your hand if you've ever tried to ride one of those.
Those are hard.
It's hard, especially if you're the person in the back,
Yes.
because you can't steer and you want to steer, but your handlebars are like frozen.
Yeah.
Only the person in front can
You're
do it.
like stuck.
Yeah, they're stuck. The person in the front's doing the steering, and so it's been my experience whenever I've tried to ride a tandem bicycle, if I'm riding in the back, I can't do it very well because I have to trust that person in front to know where he or she's going. Yeah, tandem. Okay, so here's what tandem means. Tandem, what's that mean? Like a tandem bicycle? Tandem means side by side or back to back or in single file. That's what tandem means. Actually, let me give you a cleaner definition. I'm making this up as I go. Tandem means following one another like in single file. Following one another in single file. That's what tandem means. So in a tandem bicycle, you got two people kind of front to back following one another in single file on a bicycle, tandem bicycle. You want to see what a tandem repeat is? There's a tandem repeat.
Oh, no.
Yeah, see, bicycle built for 4. So you've got not just 2 units, one right after another, but you've got 4. That would be like, that's a tandem repeat.
There's no way that works. You can't turn a corner with that.
Not very well. You'd have to turn wide, wouldn't you?
Yeah.
I think it's a real picture. I don't think it's been doctored up. I don't think it's
There's
fake
it doesn't
news.
make much sense to me.
What?
It doesn't make much sense to me.
No, it doesn't. It looks kind of... Yeah, they couldn't turn real sharp. The family that bicycles together stays together, right? Tandem repeats. Okay, in genetics, what are tandem repeats? Let me show you. Looky here. 21NT, that stands for 21 nucleotides. We're going to call that a repeat unit. Here it is. Now, you guys could count them if you want to. Trust me, there are 21 letters there. 21 nucleotides. Let's call that a repeat unit. Okay? And that's one repeat unit. I'm going to put parentheses around it. Now, look at person A. Here's person A's DNA, starts here, ends here. And inserted within this sequence of DNA, there are 1, 2, 3, 4, 5, 6, 7, 8 of those. 8 repeat units. One after another in tandem. You with me? Now guess what? In this particular sequence of DNA, the same sequence of DNA in different people are going to have different numbers of these repeat units. The number of those repeat units is going to vary from one person to another. You see why we call these variable number of tandem repeats? Make sense? This person's got 8. 8 of these, one right after another in tandem. Tandem repeats. Okay, nod your head yes if this kind of makes sense. Or is this a lot on a Friday? I guess it'd be a lot on a Wednesday, right? Or a Monday.
On a Friday when we just had 2 days off?
Yeah, that's right. So you're as rested as you're ever going to be.
No, I don't think so.
Now look at person B on the other hand. Same sequence of DNA, starts here, ends here. Look at person B. Person B has 1, 2, 3, 4, 5, 6 of those repeat units, compared to 8 in person A. Oh, let's add one other thing. If you hit that sequence of DNA with a restriction enzyme, let's say we get a cut site here and a cut site there. In the exact same sequence of DNA in person B, we have the exact same restriction sites. Here and there. The exact same restriction sites. So this isn't restriction site polymorphisms. It got the same cut sites in the same locations. What's different, though, between person A and person B? What's different? Cut sites are the same, but what's different?
There's less of them?
The what?
There's less repeats?
Right. What's different is the number of repeat units embedded in that same piece of DNA in the 2 different people. Make sense? If we look at this same sequence of DNA in Laura, there might be 15 tandem repeats. In Lily, there might be 4. In Maria, there might be 30. So you can see if you run these fragments through a gel, then this is going to be a really long fragment, that's going to be a really short one. See how you could tell the differences between people using VNTRs? Sort of. Variable number of tandem repeats. Now, check this out down here. When DNA fingerprinting is done, in order to tell the differences between people, there's something we need to know about this. Remember last semester when we were learning about DNA and I said that 1.5% of human DNA codes for proteins? 1.5%. Only 1.5% of our DNA is actually genes that have instructions for how to make protein. Okay? Like for example, my hemoglobin gene, the gene that says how to make that protein hemoglobin that carries oxygen, the DNA sequence in my hemoglobin gene is identical to yours. Identical to yours, identical to yours, identical to yours. It's the same in all of us. Thank goodness, okay? Otherwise, that protein wouldn't work. It's got to be the same in all of us. But the other 98.5% of our DNA is considered non-coding DNA. Remember the introns last semester? Introns and exons? So 98.5% of our DNA is non-coding. That means it's not genes. Only 1.5% of our DNA is actually genes. The other 98.5% is non-coding. And in your non-coding DNA, there are all kinds of restrictions, site polymorphisms, and variable number of tandem repeats. See, if you've got a stretch of DNA that's non-coding, that's not part of a gene, do you see how it wouldn't matter if there are differences between my DNA and your DNA in that particular location? Because it's not part of a gene. And so over many generations, we accumulate all these point mutations in our non-coding DNA because it doesn't matter, because they're not genes. You wouldn't want to have a point mutation in your hemoglobin gene because it could cause sickle cell anemia, right? Or something like that. But if you have point mutations scattered throughout your non-coding DNA, it doesn't matter. It doesn't matter because it's not genes. So because of that, when they do DNA fingerprinting, they target. When they do this DNA fingerprinting to tell differences between people, they target those non-coding sequences. That's what they target. And so, like in this example, person A, this is a non-coding sequence. It's not part of a gene. Happens to have 8 repeat units. Person B's got 6 of those repeats in tandem. And that's a way you can tell those people apart. Okay. Now then, let's take a look at a video clip. And this is just another person talking other than me. So you're going to hear it like from a slightly different angle. Okay? Let's check this out.
Drawing out the facts, the Naked Science Scrapbook. Hello and welcome to the Naked Science Scrapbook from the Naked Scientists. This time we're answering the question, how does DNA fingerprinting work? If you've ever seen CSI, or one of those reality talk shows where a man demands a paternity test for a baby he doesn't believe is his, you'll have seen the 2 most common applications of DNA fingerprinting. But what's actually involved in the process? First, let's look at how DNA fingerprinting was discovered. On September 10th, 1984, at 9:05 in the morning, Alec Jeffreys had his eureka moment. His team at the University of Leicester had been working for 7 years to see if it would be possible to tell people apart and whether they were related using their DNA. They'd been looking at regions of the human genome called VNTRs, which stands for Variable Number Tandem Repeats. These can be thought of as a repeating string of genetic letters or bases, a bit like the same word written down many times along a line. In this case, the words are between 10 and 80 bases in length, and the numbers of repeats of the word can be as high as 30. You inherit these repeated sequences, along with the rest of your DNA, from your parents. But because the VNTRs occur within parts of the genome called non-coding regions that aren't actually used to encode proteins, genetic changes called mutations and duplications, which are copies of the DNA sequence, take place within the VNTRs much more often than in other parts of the genome. This means that a person is likely to have a similar number of repeats to their close relatives, but people more distantly related to them and complete strangers will have a totally different number and therefore different length VNTRs. It's these differences that can be exploited to tell one person from another genetically. On that morning in 1984, Jeffreys was looking at the results of an experiment they carried out to see if a probe they'd created to pick up one particular VNTR sequence could show up variations in the length of the sequence repeats in the DNA of his lab assistant and her parents. A clear pattern was visible, confirming that she'd inherited repeats of a different length from each parent, and the world of DNA fingerprinting was born. After this breakthrough, Jeffreys' lab was the only one to offer DNA fingerprinting, and they were in high demand with cases from all over the world until the process was commercialized in 1987. Nowadays, rather than VNTRs, DNA fingerprinting makes use of sequences called short tandem repeats, or STRs. These are similar to Jeffrey's VNTRs, but are smaller and stand up better to the degradation of DNA over time, which makes them much more useful for investigating crime scenes. So let's look at an example of how DNA fingerprinting is carried out today. The DNA from a sample such as a mouth swab, blood sample, or a swab from a crime scene is extracted and purified. In order to isolate the short tandem repeats, a procedure called PCR, or the polymerase chain reaction, is carried out on the DNA.
All right. We're going to save PCR for later. Okay? It's a newer way of doing DNA fingerprinting, a newer way than variable number of tandem repeats. And we're going to put that off until later. Okay? When you guys do your own DNA fingerprint, the lab, where you do your own DNA fingerprint, you're going to be using this technique that she referred to, DNA fingerprinting based on PCR. So we're going to learn about that later. But I don't want to leave VNTRs just yet. Okay? So this DNA fingerprinting can be used in something called pre-symptomatic diagnosis. What's that sound like? What's that mean?
Before you start showing symptoms.
Excellent. Diagnosis of a disease before you start showing symptoms. Can you think of an example of a genetic disease that doesn't hit until maybe the person is like 35? Yeah, Huntington's disease. Now there's a test that people at risk of having Huntington's disease can have done before the disease hits, before they even know they have the disease. There's a blood test that can be done, and then they do it DNA fingerprinting based on VNTRs or PCR like they referred to in the end of that video. They can do a DNA fingerprinting test on those people to see if they have the gene or not. So, I don't know, that's kind of hard. Talk about a bioethical decision. If you had a parent with Huntington's disease, okay, it's autosomal dominant. Remember autosomal dominance? Okay, so let's say your parent with Huntington's disease has a big H, little h gene pair, big H, the Huntington's disease gene, little h, the normal gene. Your other parent's normal, little h, little h. All right, so the parent with Huntington's disease, big H, little h. The normal parent, little h, little h. What's the probability that you also have that bad gene? Big H, little h, cross with little h, little h. Raise your hand if you think you know the answer. Are you doing a Punnett square in your head? Anybody know the answer? 1/2. All right, so if you have a parent with Huntington's disease and you're 18 years old or 17, then you know you have a 1/2 chance, 50-50 chance of also having the gene. That would, can I say it, suck? Because you'd be going through your life thinking, well, okay, I'm going to be totally normal until maybe age 35. Now, I don't know about, okay, it's all perspective, I know. To you guys, 35 seems like really ancient, right? Trust me, I'm going to be 64 next month. To me, 35 is not old, okay? It's all relative, I know, but no, 35 is not old. So if you had a parent with Huntington's disease, you'd be going through life knowing that the chances are 50-50, that around age 35 you're going to suffer the same fate. Okay. Raise your hand if you would want to know, if you were in that situation. You would want to know if you had the gene. Okay, you know what? About half half the people say, yeah, I'd want to know, and about half the people say, no, I wouldn't want to know. Lily?
So say your parent has Huntington's and they started around 40. Would you be more likely to start around the same time, or does it always vary?
That's a good question. Chances are you'd start around the same time, but there's a slight chance you might start a little earlier.
Why not later?
Well, you could, as a range.
Okay.
So, yeah, it's the people who are children of Huntington's patients, they know that there's a 50-50 chance they've got that sword hanging over their head, so to speak. Okay? Hard to live with. I've read cases of people in that situation who they know that they could go into a clinic, get a blood test, within a week get the results. But they can't, they kind of want to know, but they can't work up the nerve to walk into that clinic, get that simple blood test, and then the lab, then they do the DNA fingerprinting and they check. They just can't work up the nerve to go and do it because it's like, okay, when I get that blood test, then I got to sit around and wait for a week for the results. I'm going to go crazy. It's hard. It's really hard. So it's a real life thing that's difficult for people. So pre-symptomatic diagnosis, using the tools of molecular genetics, DNA fingerprinting, to test for disease genes, like with Huntington's disease. Does anybody know who that is? Lily, you had a question.
Is there a possibility that a gene can mutate and you can develop Huntington's?
Very slight, almost not even worth worrying about thinking about. Huntington's disease is not something you have to worry about unless you have a parent who has Huntington's. Pretty rare. It's pretty. I can't tell you off the top of my head the incidence in the population, but it's like not even worth. Don't give it a second thought.
Yep.
Okay? Anybody know who that is? Do you know who that is? Who? Oh no, it's not John Mellencamp. That's a good guess.
That's what I thought.
No. This is a picture of a folk singer from the 1930s. That's before my time too, you guys. Woody Guthrie. I'll bet you, and he wrote a lot of folk songs in the 1930s, Dust Bowl era, and I'll bet you a lot of you sang some songs in elementary school that he wrote. You want to hear a recording of him singing one of his songs?
Sure.
All right, check it out. Now here's the deal. He died of Huntington's disease. Woody Guthrie. Okay? He has a son named Arlo Guthrie. Anybody ever heard of him? Arlo Guthrie? Well, he's alive today. He's also a songwriter and singer. In fact, he performed at Woodstock. Have you guys ever heard of Woodstock?
Yes.
Where a whole bunch of hippies got together in upstate New York in August of 1969. Big rock festival. And they were like free spirits, free living, anything goes. Now, people have asked me, Mr. Ruhl, were you at Woodstock? The answer is no. I was in junior high. But I remember seeing stories about it on the news. Well, Arlo Guthrie, the son of Woody Guthrie, was one of the performers at Woodstock. What's Arlo's chance of having the Huntington's gene? 50-50. But he's older now, of course. This was 1969. There he is today performing in concert. He's one of those children of Huntington's patients who said he didn't want to have the test.
Oh, yeah. I remember this.
Yeah, he decided he didn't want to have the test. He said, why go through life worrying about that?
Didn't they do a thing on 60 Minutes?
Yeah, there was a thing on 60 Minutes about him. He still does concerts today. He's much older today, of course, but I think he's getting to the age where it looks like he doesn't have the gene. He's in his 70s. So I think he's probably safe. But he was heads or tails, right? But he's one of those people who decided, I don't want to have that test. life's too important to worry about things like that, so I don't want to know. I don't want to know. All right, I think this is a good place to stop. I want to show you a video clip. We'll have to wait until next time. It kind of shows what Huntington's looks like. You're going to see a Huntington's disease patient talking. She's wrestling with the disease that she knows she has, and she's going downhill. Just so you can really kind of see how this works. And then I want to show you a couple other people dealing with this very same disease. And then we'll look at how VNTRs are used to diagnose this before the symptoms even start. Okay? Have a nice weekend.